All right. Please, if your mic is on, kindly mute your mic. We are going to disturbance. Yeah, if your mic is on, please. Okay. I don't know that whose mic is on. Okay, that could be Rita Ngozi.
Yeah, I'm trying to.
Okay, let me do that from my end.
Okay, okay, sir.
All right. So once more, I want to welcome each and every one of us to our second free workshop for the month of January. We have the first free 2 days workshop last 2 weeks. That was on tomato 4 modeling, and now we are here again on molecular modeling. So for those of you that happens to be your first time to HENDEP, you are welcome. And we believe that you enjoy your first lecture with us. So this training is being organized by HENDEP Industries Nigeria Limited, and is anchored by one of our division in charge of training, that is the School of Computational Chemistry and Biology. And I'll be glad to be your facilitator. My name is Osiago Henry Chibweze. So once again, we are all welcome from. We have participants from different countries represented here. Some of them have to stay awake till this time. Mostly those in India and those in Sweden, whose time difference is like 4 hours after. So once again, we say thank you. I'll quickly share my screen so we start on what we have today. So if you can see my screen, just do it to let me know. I believe you can see my screen, right?
Yes, sir.
Yes, sir.
Okay. I want to also appreciate our professors who are here. God has really blessed us with a lot of professors in our training, and it's a it's a good remark. Okay, why are we here? We have gathered for a training on molecular modeling. Yeah, for high-throughput screening of natural and synthetic molecules. I will do some background knowledge, some introduction that will help us understand what we are doing. Just like I always say it in my training, I don't always move into practical. I like my students to have a first-hand theoretical knowledge, which will serve as foundation to what we are doing. So from the introduction, if you look at the title of the training, molecular modeling, I think that has to deal with, let me just deal with the modeling first. When we talk about modeling, we mean designing something according to a pattern called model, building something according to a model, a standard, a pattern. So if I'm a fashion designer, so fashion modeling is building a clothing according to a pattern. Probably I saw a pattern in a catalog, and so I said, okay, please make me these garments to look like this one in the catalog. What that fashion designer is doing is modeling. So he's going to model raw material. Please, if your mic is on, kindly mute your mic. So he's going to model the raw material to look like the one in the catalog. So in this case, we are here to model compounds, molecules, so as to have certain features and properties that is desirable for a definite application. So maybe you want to apply these molecules in drug design. So you have to now model these compounds to look like drug. So if you want to apply these molecules in environmental pollution, environmental remediation, so you have to also model those molecules to fit in properly as a bioremediator. So that is the whole idea of molecular modeling. So from the slide, it refers to the use of computational method, because here we shall be using our computer to do that. So it involves using computational methods and theoretical framework. So two things are involved here. We combine our computer and the theory knowledge we know, because to every practical there was a theory that served as a backbone. So we shall be combining computational methods and theoretical frameworks to represent, simulate, and predict the structures, properties, and activities of molecules. So our job in this training is to teach you how you can use some computer-aided tools to represent molecules, to simulate molecules, which is seeing their behavior in a condition, then to predict their structures, the properties, and even as well predict the activities of these molecules. So these are the whole idea of molecular modeling. It has become an indispensable tool in modern drug. So if you have the passion for drug discovery, drug design, you cannot do without molecular modeling. You cannot do without it. So it is very important. Not only those who are interested in drug design, but if you have interest in material science, enzyme engineering, and chemical ecology, and so on, you need to learn how to model molecules. Last 2 weeks, we learned how to design molecules based on some features. And last week, Saturday, we had a webinar discussion on feature-based. So all our training this month has been centered on drug features, because if you want to have a drug, then you must be able to work with molecules that has the properties of a drug. So we don't just work with any molecules that we lay our hands with, but we try to work with molecules that are drug-like. So the whole idea of this month training, the last 2 weeks, the last week, and this third week now, is all about designing molecules. So during the Macrophore training, we did the designing where we use some softwares to draw chemical structures, to build our own chemical structures. But in this training now, we shall be doing screening, high-throughput screening. So you may be having maybe 2,000 synthetic molecules. We would learn how to screen these 2,000 molecules and filter off the ones that are not drug-like. Maybe you're interested in natural products, just like I told our participants in the 1 month, the ongoing macroprognosis training, that in natural product drug design, you need more than 1,000. You need more than 1,000 natural products, but you will not work with those thousands of compounds. So you have to screen them to get the ones that are drug-like. And that is the whole idea why we have gathered here tonight and why we also gather tomorrow. Now, what about high-throughput screening, HTS? High-throughput screening is a process in which large libraries, very important. So in high-throughput screening, there must be large libraries of chemical compounds, which we are going to test them. So these libraries are often millions. So in high throughput, you want to test a library of compound at the same time. So there might be millions of compounds. I was even limiting us to thousands. So here you start with millions, often millions at a time. Why is it always million? Because you need as much as possible. I told us yesterday in our training that in silico study, computational studies is based on data. And one of the challenges we're having in Africa is lack of data. So we need data, large number of data, so that we can screen them. At the end, pick out the ones that properly fit in what we want to do. So in high throughput screening, you have to screen, test large library at the same time with the aim of identifying molecules that show activity against a biological target. That is if you are interested in drug design. So if you are not interested in drug design, then you have to also identify molecules that show the desired properties you need, depending on where you want to apply those molecules. So in as much as you work with molecules, whichever work you want to do, you must bring in high throughput screening. Very important. Now, this is not like the traditional HTS that we know that involves physical assays, where you have to get these molecules. You have to synthesize maybe the 1 million molecules. You have to synthesize or isolate these 1 million molecules and begin to test their assay one after the other. It is too robotics. It's too expensive. So we shall be using computational tools to screen these compounds without having physical contact with those molecules. And the result you have will be approximate to the ones you're going to get if you do wet lab. So we are not going to be using our traditional HTS because it's actually expensive and time consuming, resource intensive, and all the rest. So why do we use molecular modeling in HTS? If you are screening, you must include the knowledge of modeling in your HTS. Since you want to screen large libraries of compounds, then you must screen them based on the model. So you must have a model now. You must have a standard, a pattern, and that will not be your rule. Though that pattern, that standard will not be your benchmark in your virtual screening or in your high throughput screening. So if you want to succeed in molecular modeling, then you must have a benchmark. What conditions do you want to follow while you are modeling? What conditions do you want to follow in your molecules so that I can screen out the one that doesn't meet that condition? Now, let's take, for instance, that I want to take students for a quiz and I have 500 students and I want to take those students for a quiz. I must screen them if the quiz is for mathematics. So I will screen them based on their performance in mathematics. If the quiz is for English, then I will screen them based on their performance in English language. So that is how it is. What kind of work are you doing? So that what will be your mind. What kind of work are you doing? Are you making use of molecules? Then one, you need large molecules. Then what are the conditions? What are the criteria for that for a molecule to fit in what you are doing? Those criteria will now form your benchmark to screen the large libraries. So we don't work with those libraries of compounds. We limit them. But the limiting them is not by your choice. You don't throw them away. You subject them to a condition. That condition will then filter off the ones that are not necessary. So why do we use molecular modeling in HTS? One, for scalability. So millions of natural and synthetic molecules can now be screened virtually without needing physical samples. Cost effectiveness, you can reduce the number of compounds taken into expensive lab assays. So why do you spend money testing so many of these compounds? You can just do your screening based on your model conditions and work with just the few that will be okay, that has a better result. Then speed. Of course, computations are faster than experimental screening. In less than few minutes, you are done. I remember when I had a training with Genomark December, something happened in that training where we had some natural products. Okay, we wanted to do drug repurposing. So we mined some synthetic molecules. So we set a rule to screen our compound. Do you know that despite the large library of compounds we used, none of the compounds passed the condition we decided to use for the screening? None of the compounds was able to pass the condition. So the participant was like, what does that mean? Should we throw them away? Some people decided to try to be lenient on their condition, trying to make their own condition flexible. But that is not so. Once you have a rigid condition, then if no molecule was able to pass that condition, what comes to your mind? Go and look for. Go and look for more. Go and look for more. So due to the prediction power, models can predict binding affinities and highlight specific interaction between molecules and their target. Then lastly, prioritization. Using MM in HTS helps identify promising leads early enough, focusing on experimental work on the best candidate. So this idea will help you to identify compounds that will be effective when you see them. Take, for instance, when you see a boy, you will know that he's a boy. When you see a girl, you know that this is a girl. Why? Because of the features of a girl. So the same way, when I see a molecule, I can tell you if this molecule is a drug-like molecule. I will tell you if it is drug-like or if it is not, because of models. And that is what everyone interested in drug design should aim at. You don't just see compounds and begin to use them. You're wasting your time. Somebody came to my office and the person gave us a work to do. I told the person that, see, these compounds you just gave me to work for you will not yield results. She insisted that use it like that. That is what my supervisor said. So do I have choice? I have to take the money and do the work for her. At the end, the results were very poor. Why? Because I've already knew that this work she wants to do has a model, a standard, a condition that the molecules must meet up to. So those are molecules we are unable to meet up to that conditions for the particular work she wants to do. So, and that is one of the drawback we have in drug design. even if we are still using computer-aided tools, we are still wasting our time. A lot of us are still wasting our time in screening large libraries of compounds. So these are the major molecular modeling techniques. There are more techniques like the MD and so on. But these three are the major molecular modeling techniques. So there are techniques we use in screening your compounds. So we have the structure based, just like what I shared with our participant yesterday, that in drug design now, we are always familiar with structure based and ligand based, but we now have feature based. We have feature based. We have fragment based. We have fragment based drug design. So I will not dwell more on structure based and ligand based, but we should spend much time on feature based, and that is what we are going to look at today. Then we shall now see if we have little time left to look at the hands-on. Then tomorrow shall be purely hands-on. And 2 hours for tomorrow's session shall be purely hands-on. Now we have structure based. So you can screen your molecule based on your receptor, based on your target. So you can screen molecules based on your target. So let's say that I want to work on a ulcer, and I have 1,000 molecules I want to target on ulcer. Do you know that I can identify which of these 1,000 molecules can actually interact effectively with the ulcer receptor? Now, if I still want to screen that same 1,000 molecules on a diabetic target, based on experience, I will know which of these 1,000 will fit in properly. We have a better result for my diabetes work. So when you are screening based on a known structure, based on a known protein, based on your target, it is structure-based virtual screening. So without me conducting my docking, I can know all this, because that receptor for diabetes at the active sites, maybe the protein I'm using, at the active site of that protein, there are certain features, that there are certain amino acid that will interact with my ligands. So when I look at the properties, when I look at those amino acids, their chemistry, the chemistry of those amino acids, I can tell how they react with my ligand. So we shall take time on this aspect when we come for, because we are making a training on March, 1 month training on macrophore and molecular modeling. So we shall take our time to explain this structure-based modeling, where you look at a protein for E. coli and you have 1,000 molecules you want to dock. You can know which of these 1,000 molecules we have a better binding affinity with the E. coli target. I have employed that many times, and it's working. So once I look at you, I can tell if you can fit in this target. Yes, it's until you come to that level. Once you look at the molecule, maybe I can tell that benzoic acid does not have the properties to interact with a diabetic target, or benzoic acid does not have the properties of interacting with a cancer cell. Because whatever a molecule can do is as a result of its structural properties due to the structure. Now, but when it comes to ligand-based, in ligand-based, we don't have interest in based. We don't have interest on our receptor. The receptor will not be known. We are interested in the ligand, in those compounds. But then, since we are not screening this 1,000 or more compounds based on our targets, what would then be the benchmark? Because there must be a benchmark. The benchmark now will be a reference material, a reference drug. So I want to work on breast cancer, but I don't want to screen my compounds based on breast cancer targets. I can look for a drug that has been known already to be effective for breast cancer. Then look at the features of that drug. I will extract the features of that known drug, then use those features to screen my 1,000 compounds. So I'll be looking for, among these 1,000 compounds, which of them has these features similar to my reference drug. So if I can have like 10 compounds from the 1,000 compounds that have similar features with this my reference drug, that means these 10 compounds will also possess similar capacity against breast cancer cells. And this is the idea of QSAR. But then you now employ machine learning. You bring the machine learning to play because you train the model based on those features you have extracted from the reference drug. We are also taking our time to discuss this by March. We have a 1-month training kept for March on this area. So we shall take our time to deal with this. So I appreciate that when that time comes, key into that training, and we have a lot. We have about 8 modules to be treated on the Macro 4 and molecular modeling. And part of our approach is not only on drug design, we now see how we can widen applications to both environments. But today we shall be exposing you to feature-based. In feature-based now, I don't need a ligand. I don't need a reference material. I don't even need a protein. It's just like when you see a girl, you don't need to have another girl to now understand that this person you are seeing is a girl. The same way when you see a mango, you don't need to have another mango tree to be able to interpret that what I'm seeing before me is a mango. So because you already know that this is a mango based on the features of a mango. So that same way is what we are employing in feature-based. So you will be able to know the feature of a drug. At the end of this session, you'll be able to know drug features. For you to serve as a drug, there are features of a drug. Just like the same way if you see a Christian, you don't need another Christian as a standard or reference material for you to interpret that the person before me is a Christian. You would know from their character and behavior. So that is the idea in feature-based virtual screening. So once again, welcome. Sorry if I'm really talking too much. Now, in feature-based, as I've said, we are looking at the features, what you see. So there are about 10 features that I've classified, but I will give you just 4 for this training. Remaining 6, we are reserving it for professionals who come for 1 month training. Of course, 2 days training may not give you that whole knowledge you need, but I'm trying my best to see that you go home with something. So we have classified those features. One of them is the pharmacophoric features. We discussed this extensively last 2 weeks, and that was what we used to design our molecules. We can also use them to screen our molecules. Yes. We used, and I'm really glad that some persons dared to do the mini project, and they were able to model 100 up to 100 molecules using de novo modeling. So the same way we were able to use these macrobic features to build molecules, you can also use them to screen your molecules. So what are these macrophoric features, they are abstract features that describes the essential spatial arrangement of interaction features required for molecular recognition. So very important for molecular recognition. What I'm trying to say is that macrophore are abstract features. They are properties that are required for a drug to interact effectively with a target. So these features must be there. So that means if you know these features and you have 2 million compounds, you can just write a model, although it's best done using programs. It's best done using programming softwares. You write a program that will say, okay, from my 2 million compounds, I need the ones that fit into these features. And you just see in less than a click and everything will be sorted. But because we have people without programming knowledge, or who have poor knowledge, people who don't also have knowledge of programming, we shall just use our Excel to write a function with our Excel. That will be very, 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 very easier to write a function with our Excel, and the function will be able to sort the ones that fit in to our properties. So some of these features of the drug are hydrogen bond donors and acceptors. So they have role to play. So I'll be giving you 4 classifications of features that must be in a drug. Then in each of them, I'll be giving you examples, about 10 examples, and what they do, except for this first one. So we'll give you 10 examples and their role they play. So you can now use all these features, do your screening. Definitely, you may not even find a molecule that possesses all those features. Just like a lady looking for a guy, and the lady was able to write down some criteria of the young man he wished to get attached to. At the end, you discover that none of these men has these features. So you have to create them yourself. Yes, so if you have used thousands of molecules and you have subjected them to these features and none of them was able to pass, so you have to create the molecule yourself. And it is part of what we'll be doing by March. So we have hydrogen bond acceptor, hydrogen bond donor. We have hydrophobic regions, and we have the aromatic range. So there are 4 basic. Though we have the ionizable negative ions and the positive ions. They are there too, but there are 4 major macrophoric features. The hydrogen bond. So you can screen based on these 4 properties. What you just need to do is to develop a criteria, like how many hydrogen bonds do I expect to have in my compound? How many hydrogen bond acceptor is okay? How many hydrogen bond acceptor is okay? You must have this benchmark. Then how many hydrophobic regions do I need for effective drug interaction? How many aromatic range is necessary? So once you establish this benchmark, which is what we'll be doing in the hands-on training, once you establish this benchmark, you can now screen your compounds to see the ones that passed this benchmark. So these features function collectively as interaction blueprints, independent of chemical scaffold. So independent of your scaffold, the backbone of your molecules, these features always work together. They always work together. So let's see examples of the macrophoric features and their implications. We have the presence of multiple hydrogen bond. Very important. What does hydrogen bond acceptor do? If you have a molecule that has some part of the molecules, maybe some of the functional groups there some part of the substituent can accept hydrogen, like the carbonyls, the ethers. So if you have them around your molecules in a good amount, they help to enhance specificity towards the polar binding pocket. Of course, this amino acid in your protein, some of them are polar. So once there is a presence of polar bond acceptor, it will help to accept those amino acids that can give out hydrogen. So if we have more amino acids that can give out hydrogen in a binding site, and then you have some substituents in your structure that can accept this hydrogen bond, it will make them to specifically bond together. Yeah, so it will increase that binding. So this is what multiple hydrogen bond acceptors do. Their presence in the molecule enhances specificity. So I can screen my thousands of molecules to see the ones that we possess these hydrogen bond acceptors. So when it comes to hydrogen bond donors, they increase binding orientation accuracy. So I'm not giving you their role, their implications. So that is why you must, in fact, it's a must. Do not do docking. Don't dock without screening your compounds before docking. It is irrelevant to dock without screening. And you see that if you can screen based on models before docking, then lead optimization will not be stressful again after docking. So I think let's just include this in our docking workflow. Now we have the hydrophobic regions, those ones that don't like water. What do? They help to improve the affinity in lipophilic cavities. Of course, there are also some other amino acid in that binding site that are not water loving. So the presence of your hydrophobic regions will help to interact with those lipophilic cavities or sites. We have the aromatic ring system. What they do is that they promote pi-pi stacking with aromatic amino acids. So your aromatic region in your ligands will help interact with those amino acids that has the double bonds, that has pi bonds. So it will help them to stack, to help them to interact effectively. So imagine now that you have compound that possesses these features, but you don't know them, so you have to screen them to see if they possess these features. The multiple aromatic rings, they increase rigidity and binding persistency. This will even help us reduce the issue of having compounds that are not stable in MD. When you perform your dynamics, it will help you reduce compounds that will happen not to be stable. So when you screen very well, because we have been told, not just we have been told, we know that a high binding value is just a guide. It's just an insight. It's nothing. It's just an insight. If you don't screen, you may have high binding value, but cannot be used as a drug. So that is the essence. The presence of heteroaromatic rings introduces both aromaticity and hydrogen bonding. All right, are we together?
Yes, sir, right here.
Yeah, so just be taking
Yes, sir.
the session. Yeah, thank you. So I just taking my time to do this. We don't have hands-on training. But before the hands-on training, you must be grounded in what you are doing so that you can do by yourself, or you can do whatever you want to do by yourself. You can be able to manipulate it because you already have a foundation. You know, the building is not the problem. The problem is the foundation. So we have a lot of persons who have heard about docking and they love docking, but they don't know what those things are all about. So the
Sorry,
same way
sorry,
we
I've not explained the last one, hetero something.
okay, heteroaromatic rings. What about heteroaromatic rings? We have those aromatic ring that contains atoms other than carbon, like the pyridine, the furan in a ring. You know what usually make up that if you look at a benzene ring, is 6 carbons at the node. But if I decide to remove one of the carbon in that ring and introduce nitrogen, that becomes a heteroaromatic ring. They are classified as heterocyclic compounds. For those who have little foundation in chemistry, they are heterocyclic compounds. So the presence of these hetero compounds in your aromatic ring, they will help to introduce both aromaticity and hydrogen bonding. For the reason why they will bring in hydrogen bonding is because if you look at most of these heteroatoms, they are mainly the non-metals. They are mainly the non-metals who are ready to accept hydrogen, like nitrogen, oxygen, then the TOs, which are the sulfurs. So that is why we are saying that the presence, the presence of heteroaromatic rings will help to introduce both aromatic stability and hydrogen bonding. All right, thank you. Now, away from macrophoric features, is just one of the features. Another feature that you need to also incorporate in your screening is functional groups responsible for activity or toxicity. Yeah, I said this first week of January in our macrophore training. I said that the chemical behavior of a molecule now lies on functional group, because functional groups determine the chemical reaction. How a molecule will react is based on the functional group. So a molecule without functional group invariably cannot react. So how a molecule will interact with a receptor, you know, this docking we know because we are familiar with docking, so we may not even look at docking. This docking we know is a chemical reaction, is a chemical interaction. And since it's a chemical interaction, it means that the molecules of the protein called amino acid will react with the molecules of we react with the molecules of your ligand. So let me put it that way. The molecules in your protein, which are the amino acid, will now react with your ligand, with your own test molecules. So it's usually a chemical reaction. Maybe we should start learning how to balance the chemical equation in a docking process. Yeah, we start learning how to balance the chemical equation because it's a chemical reaction. Now, because it's a chemical reaction, it involves the functional groups in your molecule because they actively interact with the amino acid. So functional group has a role to play in the activity, not just in the activity, also in toxicity. I mentioned that I don't even build molecules using a nitro group, NO2, because that NO2 group is toxic. So I'm taking my time to preparing a material on functional group, the activity, and toxicity. So it was from that material I'm preparing that I have to lift some of them for this training. That functional group has a role to play in your activity and in your toxicity. So I'm into medicinal chemistry, but I have this advanced knowledge in synthetic chemistry. So this will really help us in synthesis to build molecules, then synthesize them. Molecules that we don't need to be stressing ourselves looking for their toxicity. All right. So functional groups determine chemical reactivity. Very important. I've already explained that. It determines the chemical reactivity. So you can screen your compounds based on their functional groups. It determines the metabolic fate, the binding interactions, and toxicity risk. I shared with us that in a pure drug, we have been able to discover that a drug can be toxic in two ways. It can be toxic based on the structures, based on the functional groups in that API. It can also generate toxicity in the body through off-target interaction. Because while you are trying to inhibit an enzyme, you may end up activating other enzymes which may not be good. So why that of the in situ, which the one that happens inside, where I'm activating one enzyme, another enzyme will be inhibited and then leading to a side effect. Why that is there and inevitable most times, we must now do our part to reduce the toxicity based on the structure. So we must do our part because that one is physical. It has to do with us. Some groups act as activity enabling moieties. So some group can actually boost activity. Yes, some functional group does that. At the same time, others may function as toxicophores. I believe that there's some of us who are studying toxicology already. We know more than me in this aspect. Yeah, they will know more than me in this aspect. So now, that scenario where some functional groups we boost activity either as inhibitor, as activators, or modulators. At the same time, too, some functional groups there will help boost toxicity. That will then introduce adverse drug effects, adverse vascular effects. So you can screen out those compounds that will be toxic. You can screen them. You can use your ADMET properties to screen out the ones that may probably lead to genotoxicity, hepatotoxicity. You can screen them out, and we'll do that. I assure you, we'll do that in this training. The feature-based drug design focuses on identifying, retaining, modifying, or eliminating functional groups based on their biological consequences. So we can retain. I will rather use these two words for now: retain or eliminate, because we are not going to focus on modifying, but we are going to identify first. Because modify has to do with using your structural softwares. So we are going to identify them. Then we now retain the ones we need, eliminate the ones we don't need. So you can screen based on macrophobic, join with functional group. My God, is well. So examples of these functional groups and their role, their implications, so that you will know as a drug scientist. So you will know. One is primary amines. The primary amines are the NH2 groups. What is their role? They help enhance ionic interactions, but increase metabolic vulnerability. So that is, I've taken my time to study amino as functional groups. I've taken my time for 1 year to study the chemistry of functional group in drug design. It's a test, though we are still writing that test. The chemistry of functional groups in drug design. So the primary amines. Now, what we discovered this from studies. We discovered from studies, a lot of studies. We have done some systematic reviews, and we're able to discover the role of these functional groups in most reports. So we are saying this out of reports, out of works that we have consulted. So primary amines enhance ionic interactions. Of course, they help interact. In fact, the presence of your primary amine in any compound will give rise to a conventional hydrogen bond interaction. So now you see that these some of these functional groups, they are related, they have role to play in the macrophobic features. Like the primary amines now, we have a role to play in the hydrogen bond donors. So you see that they have a role to play in hydrogen bond donors because they have to donate their hydrogen since they have NH2. So they can now donate the hydrogen. Then we have the secondary amines. Yes, we have the secondary amines. They balance binding strength and metabolic stability because they don't have much hydrogen to release. So because they don't have much hydrogen to release, they just have one hydrogen, NH. So we have two bonds to the nitrogen and then one H. So because of that, they help to just balance. They are not really hydrogen donors. They barely do that, but they help in stability. They help in stability. Then the hydroxyl groups improve solubility and hydrogen bonding. That is clear. The hydroxyl groups helps to improve because of the OH. So this drug can really be absorbed, except the drug will be used for neuroprotective for neuroprotection or for neuro disorders. Then such drug may not permeate the brain barrier. So for a drug that will be designed for the brain, that means it must increase the lipophilicity of that drug. But otherwise, the hydroxyl groups in your compound will improve solubility and hydrogen bonding. The carboxylate groups enable strong ionic interactions but limit permeability. So when you have this carboxylate group, like a carboxylic acid group, the esters group, those ones as a result of COO, where you have to remove one of the H, like COOH, then one of the H will be replaced. So they are the carboxylate group. They enable strong ionic interactions. So they are needed. The sulfonic groups increase polarity and enzyme affinity. I will be so fast so we can meet up with time. So if you can understand the chemistry of these functional groups, it will also enable you in not just screening, but in designing. So while we learned about designing last 2 weeks, we only learned designing based on macrophobic features, but you can design based on functional groups knowledge. The halogen substitutions enhance metabolic resistance. Because these halogens, they're also related to our macrophobic features because they play a role in the hydrophobic region. The halogens now plays roles in the hydrophobic region of your molecule. The ester linkage enable controlled metabolic activation. And this is also similar to the carboxylate groups. The thiol groups, the esters, the one that has the thiol groups, the one that has sulfur, they facilitate meta coordination. We have put up a training for this year, but I don't know the month where that training is coming for nanoparticle, nanotech, nanomodeling. I don't know the month. And also we have a training for meta complexation. So compounds that have thiol groups can facilitate meta coordination. Yeah, so there's a training for this, for meta coordination, because we're actually driving towards bioinorganic studies, where we have to join organic molecules with inorganic molecules to form a complex, and that will be so effective. The opposite functionalities increase reactivity and toxicity risk. So please, those without chemistry background, I'm sorry. I have to apologize. Those without chemistry background, this is to show you that you need chemistry in all your discipline. But I'm really sorry. The opposite functionalities increases reactivity, but at the same time, increase the risk for toxicity. So when you have compound that has a opposite, just be aware of this. The nitro groups contribute electronic effect, but poses mutagenic concerns. It can lead to mutation. I've thrown a little light on these nitro groups, where I said I will never design a molecule for a drug that has a nitro group. So you can see now that they have a mutagenic concern, so they can cause mutation. But I think we are coming towards an end for this presentation, so we can do a little hands-on. Then, while tomorrow, 2 hours tomorrow is purely hands-on. Another feature that we look at in modeling is the molecular weight, so the molecular shape and flexibility. These features are overlooked. So here is my question. How many of us screens our molecule based on these features before docking? So if you don't practice that modeling, that screening before docking, please do that. It will help you. It will really, 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 really help you. Everything I'm teaching is always based on my experience, based on what I have been doing. So most times I teach what I do. So if I discover a new idea, I love sharing the idea to people who may listen. So, and that is the best thing. That is the best thing. So molecular shape gives or governs geometric complementarity, while flexibility determines adaptive binding capability. Now, the shape of your molecule is very important. The flexibility of your molecule is also very important. Most of us must have heard about these features called the number of rotatable hydrogen. We have seen it in some ADMET results or in some physiochemical properties. Number of rotatable hydrogen, flexibility. But we don't pay attention to those things. So all we know is let you lie down, do your docking, do your MD, get a result, publish as well. So the shape, which is the geometry of your compound, is necessary. We shall look at their role. Excessive rigidity reduces adaptability, whereas excessive flexibility increases entropic penalties. So what I'm saying is that you must balance their rigidity and the flexibility. Of course, if your compound is flexible, it can easily adapt to different binding sites, to different poles. It can easily interact. It can easily work around the receptor if it's flexible. But at the same time, if it is too flexible, it means it will lack specificity. If it is excessively flexible, it will lack specificity. It is like jack of all trades and master of none. Now, successful drug design achieves controlled conformational adaptability. So conformers, this is where we have conformers. You know, we hear about conformer, but most of us we have not even paid attention to what are these conformers. What are these conformers? We have 2D conformers, okay, 2D structures, 3D conformers. Do you know that a molecule can have more than one conformer? Most times, you can have one molecule with 10 different conformation. What does that mean? 10 different shape, different molecular arrangement, but the same molecule, the same position, but the shape is totally different. So you can even do a docking based on conformation. That is nice. You can dock one molecule, but with different conformations to see the best conformations. I think I've not tried that, but I think since it just kept my mind now, so I will note it and try it. If it works, I will share it with you. Now, what are the examples of this shape and flexibility and their role? Rigid ring system enhance binding precision. Like I said, that if your shape is too flexible, that means it will be jack of all trade and master of none. It can penetrate anywhere. There is no specificity. But if the ring is rigid, then it will enhance binding precision, specificity. Now, flexible linkers allow induced fit interaction. It can easily penetrate, can be induced in that target. So if it's, but not too flexible. So you can screen based on these criteria. Microcyclic frameworks. Yeah, microcyclic frameworks combine rigidity and flexibility. Linear molecular geometry facilitates deep pocket penetration. So if your molecules can be linear in geometry, like 180 or planar, then that will be better. The branch structures improves selectivity. So that means if you're designing your molecules, I'm putting up a training, but I don't know when that will be, on de novo modeling. How to actually on your own build drug, design drugs that will be more effective than the ones in the market. You can just do that. You just sit down and listen to these things, combine them together, build your Jesus. You can build a Jesus, a drug that will act like Jesus for us, a super drug that will be very much more effective. The only challenge you may have is possibility of the synthesis. Conformational locking improves binding persistence. So I don't want to be so fast. Shape complementarity reduces off-target interactions. Flexibility modulation controls multi-target activity. I've thought about that. Shape asymmetry improves receptor discrimination. So when we get to match, we shall deal with this shape and flexibility of drug. We shall take our time to look at them very well with hands-on practice. So we'll just look at some of the things we have discussed today and then practice them hands-on. Then lastly, this is the last feature I will explain or be given to us, the physicochemical features. Most of us that have done Swiss admin, we use Swiss admin, we use admin lab, we use other kinetics tools. We usually see these physicochemical properties. Even when you are downloading your ligand from PubChem, you do see these physicochemical properties, but what do you do with them? You often overlook. You don't know that they have role. They have role to play in absorption, distribution, permeability, elimination, and environmental fit. They have role to play in even, in fact, they have major role to play. Feature-based drug design optimizes these properties to ensure therapeutic efficacy while minimizing toxicity and environmental persistence. So if you can actually use feature-based drug design approach to optimize these properties. Optimizing means look for the molecules that has better histochemical properties, drug-like. We must have heard about drug models. These drug models we are made from these features. You know, drug model is a feature-based approach. Drug-like models, drug-likeness models, lipid, Weber, Egan, they are based on these features. So you can create your own model. You can name it like one person calls me Henko. So I can just create my own model from just pick some of these features and call it Henko model. And that would be good. It might be acceptable and included in what we are doing. So examples of physicochemical properties and their implications. Here we have the lipophilicity. So it improves membrane permeability. I've explained that. Excessive lipophilicity increases bioaccumulation risk. Like I talked about the blood-brain barrier. It is not good for your drug to permeate the blood-brain, except that drug will be used as a neuroprotector. Yes. Hope we are together.
Yes, sir.
Yeah.
Yeah, we can hear you.
Yeah. So except that drug will be used for neuroprotection. I'm currently doing a work for a master student on Alzheimer's disease. Yeah, on Alzheimer's disease. So that means we are going to model the compounds to be able to have these properties. You know, for you to penetrate the blood, the blood is more of lipids. The brain, sorry, for you to penetrate your brain is more of lipids. So whatever that must be used for the brain must have that lipophilicity properties. So, but if it's excess, it will increase accumulation risk. It will really lead to some high side effects. Now, balanced polarity enhances solubility and target access. So, make sure that your physicochemical properties not just excessive, not too much hydrophilic and not too much hydrophobic. So make sure it is balanced. Make sure it's balanced. So you can get that from your solubility parameters, your log P and other parameters. It can help you. High polarity limits passive diffusion. So it's not balanced. Low molecular weight improves oral absorption. So if we have a compound that has a lower molar weight, it can easily be absorbed. That is why lipid food will limit us to not more than 500 grams per mole. Now the molar weight shouldn't be more than 500 grams per mole for your drug. You can screen your whole molecules based on these properties. Another example is high molecular weight necessitate targeted delivery. Now, since low molar weights, like we just explained now low molar weights improves oral absorption. Now, if you have a high molar weight, if you have a high molecular weight, then it means that that compound may not be absorbed readily, but we can now use it for targeted delivery. We are preaching two things currently now in drug design. We are preaching precision medicine and we are preaching targeted delivery. So I don't want to produce a drug that can go to any site. You can produce a drug that will be specific for only one site. That makes it effective. And one of that is giving your drug high molar weight. So that means such drug will not be taken orally. Because for a drug to be taken orally, it must have a lower molar weight. Controlled surface area, working with your TPSA, it helps to optimize your bioavailability. Charge balance, the electronic charge minimizes non-specific interactions. So I'm just taking us, the physicochemical compliance improves clinical success rates. So what that means is that if you can comply to these physicochemical properties, then it will have high success rates. High success rates. Environmentally optimized properties reduce ecological impacts. So if you are designing your molecules to be used in the environment, then if you can optimize them based on the desired properties for what you want to use it for, it will reduce ecological impact, like risk. So in conclusion, feature-based drug design relies on the precise understanding and manipulation of molecular features. That is what the whole idea. You must understand, must be able to understand and know how to manipulate the molecular features. Macrophores, functional group, electronic and steric properties, molecular shape and flexibility, and physiochemical parameters. You must learn how to manipulate on these and more. At least this is enough. So as you are living tonight, what should be your mind is, how will I be able to manipulate these macrophore properties, the functional group properties, electronic and steric properties, molecular shape and flexibility, physiochemical properties. How will I combine them together in one screening? So that will be what will be in your mind. If you can achieve it, glory to Jesus. We are meeting my match to open you up on how we can do that. So by focusing on features rather than entire molecules, very powerful. Focus on features rather than entire molecules. Modern drug design cannot achieve greater precision. So the reason for failure, we always preach 90% failure and 10% success. And that may discourage an early career scientist who want to venture into drug design. But if you can focus on features rather than your entire molecules, what is doing the job is not even, is the features of those molecules that is doing the job. What makes me a girl is the features you see in me. What makes me a boy is the features you see in me, not the entire whole, not the human being, not my internal system, but what you are seeing outwardly. Of course, we know from biology that the genotype determines the phenotype. So let's focus on the features and not the entire molecules. If we can do this, that means we can achieve greater precision, safety, efficiency, and sustainability. And this will even help us to position our compounds at the forefront of future biomedical and environmental innovations. So once more, I want to appreciate you for staying to this point. We are going to do a little hands-on. Yeah, in the next 40 minutes. So in the next 40 minutes, we shall be taking some hands-on training and what we have just said now. And tomorrow we'll continue from hands-on training. So at this point, let me take your questions. Yeah. Let me take your questions. Let's interact a bit before we move to hands-on. All right. Yeah. I'm seeing Musilu Ayofe. Yeah, go ahead, Ayofe.
Thank you very much, sir,
Thank
for
you so
this
much.
beautiful presentation. Firstly, I would like to suggest that maybe the questioning segment should be after the little answer, so that the question will not take much of the time. So that at the end of the day, we'll be able to enter the answer. So that on that basis, I want to reserve my questions after the answer.
Okay. Okay. So I will go to. Yes, I support Ayofe, Professor Ayofe. I support Prof. Yeah. So quickly, before us now are two approach. In fact, there are two focus we have based on the title of the training: screening natural products and screening your synthetic molecules. What I will do is that I will start with the synthetic molecules because we can assess them. But natural products, how can we achieve? I want to ask you, how can you achieve up to 1,500 compounds before tomorrow? Before 8 p.m. tomorrow. Can you do that?
It's communicating.
Can you
Wow.
can you gather up to 1,500 natural products before tomorrow?
That seems a bit difficult, sir, because we are here to complete the assignment of Pharmacopho.
Okay, I will provide it for you for the training. I will provide it. Yeah. That is better. I will provide it now.
I think it depends on the mode of the assignment and how to do it, and probably the software to be used or the skills involved.
We just want maybe for a particular plant, or maybe not even for one plant, because the idea first, I just want to get the idea. The idea is just screening natural products. So if you want to limit it for a particular plant, fine, screening the natural product, or you can combine from different plants. We just want to stabilize the idea of screening natural products. So after when we have created the idea of how to screen natural products in your own practice, you can now streamline your own to a particular plant you are working on. All right. But even if I want you to focus on a plant or different plants, it will still be difficult between now and tomorrow to achieve 1,500. So what I will do is, I have a lot of database I have created. I will just merge all the natural products and make sure we have up to 1,500. Then I will send it to you. I will send you the file tonight so we can use it for tomorrow. So that means tonight I will focus on synthetic molecules. Hope that is okay.
Okay, sir.
Yeah, but if you still want to, if you can achieve by yourself this 1,500, I'll be glad.
The natural products you're referring to, these are bioactives now. Bioactive compounds from plants.
Yes, bioactive compounds from plants.
Okay.
So let's look at one of the chemoinformatics database. Okay, let me share my screen again. Before I hold on, somebody sent a message. Okay, thank you very much for this powerful. All right, thank you. So let me share my screen. It has been a wonderful session. So can you see my browser?
Yes, sir.
All right, so if you are with your system, let's go to PubChem. Let's go to PubChem. So it's hands-on. You follow me. All right. So if you have not made the commitment fee for your certification, I will advise you to make that payment. We'll give you a grace till tomorrow morning, so we can prepare your materials, because you have access to your materials. We are providing about 5 textbooks on molecular modeling. We are giving textbooks for all our participants. Then we are giving your summary notes, like a summary of the class. We have access to the videos. Then we are giving you some journals some published articles on this. So you see that what you're learning is feasible and it's practice, you can practice it. So you can make that payment tonight so we can have access to the videos by tomorrow morning. All right, so here is PubChem. We are very familiar with PubChem. So what am I going to do here? I want to use PubChem to retrieve some molecules. Now I will need them because in virtual screening, you screen based on their SMILE code. We use their SMILE code to generate these properties that we need. So even if we have them in structural form, in structural data, in SDF, we can still convert that SDF into SMILE code. So have we been able to download have we installed the Open Babel for those who don't have it?
Yes.
Yes, sir.
Okay. Because
Yes, sir.
we will use it to convert structures, if you have your structures, to SMILE code. Yes, to SMILE code. Then from that SMILE code, we can now do whatever I want to do. So let's say I want to work on drug repurposing. Okay, let's say I have a drug, like I have aspirin, for instance, and then I want to modify aspirin, but I don't want to do that by myself. I want to get similar structures of aspirin, then screen them to see the ones that are drug-like, then use them for my study. This comes as lead optimization, because after your docking, you identify your heat. That heat now, you optimize the heat by creating analogs of that heat. Then we test the analogs to see the one that might be better than your heat. So we are assuming aspirin is a heat, it's already a drug. Now aspirin is already a drug now.
No, sorry, I want to confirm. See my network.
What's that?
Hello?
What is the issue? Can you hear me? Can anyone hear me?
We can hear you.
We are
Can
here.
hear you, sir.
We are together, sir.
All right, so that is your end. So aspirin is a pain relief, is an anti-inflammatory drug. So, but I want to now produce another. I want to produce a better. I want to see if I can have a better aspirin derivative that might be better than aspirin as a pain relief. So I will need analogs of aspirin, and I don't want to draw them myself. So I can lift those analogs from PubChem based on similar structure search. I explained something similar to this yesterday in our macrognosy training. So let's work with aspirin for now. So you type aspirin. Once you type aspirin, you search. Remember, I've explained what I want to do. I want to look for synthetic molecules that resembles aspirin, that has maybe that same aspirin scaffolds, but with different substituents. So there might be 2,000 or 3,000, 5,000, but I don't want to screen these 5,000 at the same time. So I can now subject them to my model and test them to see how they can see how I can filter them and see the ones that will be drug-like. So once you have searched for aspirin, The first one that comes up is your best match, exact match. We can call it exact match search. Now, below the exact match search, you will see similar structure search. So click on the similar structure. Okay. So click on similar structure search. You can see my cursor where it's pointing at. Once we click on the similar, thank God for network tonight. Yesterday, the network wanted to be very bad. That was when I promised to be your president. So once we click on similar structure search, you are seeing identity, which is just one, showing us that it is only the aspirin, the best match we choose. Similarity, there are 6,900 compounds that are closely similar with aspirin based on fingerprint search. Of course, this fingerprint uses the Tanimoto principle. Time will not permit me to teach you how to calculate the Tanimoto principle to show how to use it, but it has been built already in this pop game. So we are having here 6,903 compounds that have similar structure, in fact, that have similar structure with aspirin. If you scroll down, you will see that all these structures maintains the aspirin scaffold. This is the aspirin scaffold. So you see that all the structures you see here, scrolling down, maintains this same aspirin scaffold with little variation in substituents. Look at them carefully, with little variation in substituents. So we are going to download them. We are retrieving these 6,903 compounds. When we retrieve them, we'll now conduct a virtual high-throughput screening on them based on the model we just learned. So it's a very simple thing. This kind of work is really done using machine learning. In case you want to do a high impact publication, high good job, you can screen with machine learning. But even if you screen with simple Excel, due to machine learning needs little programming too. So if you want to organize a class for machine learning, that means we'll have a 1 month class where we'll just groom you from scratch, so you won't get lost. But you can use Excel for the screening. So all of us are familiar with Excel, just a little function for it, for the screening. So let us now download them and retrieve these 6,903. It may take time to download though. If I see that it's taking time, I will look for another derivative that may have smaller number of similar compounds, so it will be easier for us to work on. So what do? You just come to download. This download here. Go to download here. Click on this drop down arrow pointing down. Once you click down, so there are two ways we can do. There are two things we can do here. The simple approach is to just download them as CSV. If you download them as CSV, you will not have the structures. You only have an Excel file, a file that can be opened in Excel containing the CID number and the SMITE code with other information that we may not need. So when you download a CSV, you have the SMITE code. Of course, it is the SMITE code that we are going to use to generate the properties we need. But for the sake of learning, we are going to download the structure because we need the structure. The reason why I love downloading with structure, if you download with CSV, you just have the smart codes. So after you screen, so what now do you, will you use the smart code for docking? You then need the structure. So you have to go back and start downloading again their structures. But if we download the structures as SDF, we can now convert the structures to smart code. So that means we have our structures, we also have our smart codes. But if we download the smart code, we may find it difficult to convert that smart code to structure at once. But we can easily convert our structures to smart code. So we will download our structures. So what you do, change this 2D to 3D. Now, this 6,903 compounds you are seeing here comprises of both the ones that are 2D and 3D. So I'm just interested in the ones that are 3D. So I will just pick 3D conformations. Your number of conformers, like we just explained about conformation. Pick one. So I just want to work with one conformation. The one that, so it will download with just one conformation. But if you pick 10, it means that each download, each compound that downloads is already carrying 10 conformers. So I don't want to do a work on that. So just want to focus on one conformations. Then the next thing we do is to click on SDF. Hope we are following together.
Yes.
Yeah.
All right. So I don't want to be so fast. I want to take the time. So then once you click on SDF, it will start to download. Now, they will not download separately. They will download together in a single file. Yours might be faster, but mine is still
Come
downloading.
again, because I lost due to network. Number of conformers, 1, right?
Yes, one.
Mine have been I've downloaded already.
Jesus. So you have a very good internet.
So man, downloaded already.
My God. So me, I'm just here waiting for you people. All right. So let's see. It will soon download. So let's see. Does that mean my internet is poor or my RAM is slower?
Mine has downloaded. It's about 29 MB.
It's very small now, so it will just download. So once so it will download as you have a single file. Now that single file you see contains no longer the 6,903, because most of them out of this 6,903 might be 2D files. So because we selected 3D files, so it will just give us the 3D files. Yeah. Why will my participant download before me? Is it not an error? My internet is okay. I don't know what's happening. I downloaded this thing before and it went so fast. Okay. What am I going to do now? What am I going to do now? If I pause and play. Okay, let me just look for the one that can download faster with me. Oh, I want us to use the same thing. What
So
kind
it
of
will soon
network
be
is this?
done. At
Eh?
least it's almost half. I said it will soon be done. It's 29 MB. Yours is about 14 MB now. So it will soon download on your end.
Okay. Well, let's be hoping. So why that is going, as every one of you let me know if I have my partners here that they only see downloading.
Mine has
Oh,
downloaded.
I'm left alone. Jesus Christ. Okay.
Not alone, no.
Okay. I'm not alone, right?
You are not alone, sir. My own
Oh,
is not
thank you,
downloaded.
Jesus.
You are not alone.
Glory be to you. So why that is doing, let me keep explaining something. So now it's coming as a single file. Let me tell you something. If you are doing docking with Pyros and you are docking 50 compounds at a time, please learn how to cluster your compounds using PubChem. So using a Open Babel. You learn how to cluster your compounds into one file. Like now, you can imagine all these compounds are in a single file. So if you are docking, once you just open that file, all the compounds in that single file will also open. If you try to open that your download now, for those of you who have downloaded your own, if you open that download in Discovery Studio, you will see all the thousands of files will open, but they're in a single file. So that is the beauty of this. You can also do that with Open Babel. So while we are waiting for you to download, let's launch your Open Babel, because that is the next thing. That is the next stage we'll go to now. Open Babel. So launch your Open Babel. While we are waiting for it to load, you can launch your Open Babel.
Please, sir, is it an app? I have not downloaded it too.
Yeah, while you are waiting for it to download, launch your Open Babel. Me too is downloading. Mine is still downloading. So
No, it is the Open Babel I'm asking. Is it an app?
hey, Jesus, did you receive our mail?
Yes, I received it yesterday, something like that.
Hey,
But
if you
today
received
is
it,
my first graduation.
today the first time here.
Yeah, today is my first
Congratulations.
class.
Okay. In that mail, we provided the link to download Open Babel.
Okay. Let me quickly see if I can do that.
Oh, God. Sorry, so sorry. Because it's there. In fact, it was very bold, though. The link was very bold. Yeah, we say, you see,
Very, very, very bold.
Okay,
yes,
I
you
can
see,
see it. I can
click
see it
here to
now.
download Open Babel.
Yes,
I
yes.
made it
Let me
very
do
bold
that.
so that everybody can. All right. So those that have it, let me to God. I've left that category of
I
downloading
can't
in
see your
progress.
screen. I'm only seeing HD look.
The patient will get the first one.
Maybe we want that will kill you. That's your adage it has been it's no more what a patient does not eat the fastest food a day. Maybe we want that will kill you.
Is the presentation still on?
Sir, you said? So I'm opening my Open Babel. It should open. Just a lot of things are in my system, and even I have some program running right away, so it might be a bit slow. Okay, then this is the one. Okay, let me remove this one. This is the recent download. Okay, let me remove these ones here. Let me delete them. I was using it for somebody. Okay, all right. So this is Open Babel. This is Open Babel. Can you see my screen?
Yes.
Yes, sir.
Yes, sir.
So what we are going to do here,
Sorry, can anybody hear me?
yeah, I can hear you.
We can hear you.
So what we do here, there, please, if your mic is on, kindly mute your mic. Now, there are a lot of things to be done here. OpenBabel has a lot of features, but I'm not going to teach about OpenBabel. I'm just teaching you how to use it for file conversion, converting from one file format to another file format. That is just one of the use of OpenBabel. There are a lot of things. We have a training on bioinformatics. So on that training, we'll teach you more about chemical softwares and databases, so which OpenBabel is among them. You can use OpenBabel to convert your 2D to 3D. So those in the macrosy training, I mentioned that if you download some compounds in 2D, you can use OpenBabel to convert them from 2D to 3D. You can use OpenBabel for clustering. You can use OpenBabel for fingerprint search and others. So, but just focusing on what we want to use it for. We want to now convert the aspirin derivatives. This can form a paper for you. You can just develop a paper, tag it for those who love paper publication. Because I'm not a fan of paper publication. So those who love paper publication, you can just design a paper and call it molecular modeling. Just find a name, modeling, docking, and so screening of aspirin derivatives. Or so you can form it, and it's a good paper. But while you are publishing, please let this be in your mind that you are publishing your paper for products. Let it be in your mind, though. I did a crazy work today. In Nigeria, I won't call the person's name, but we know. I searched the #1 researcher from the new list now we are having, the top 500. So I looked at the #1 in Nigeria. My interest was, how many products does this prof have? I know him very well, though. How many products? It will amaze you what I saw. But that is by the way. Now, for you to use OpenBabel to convert your structures to SMILE code, that's the next stage. What you will do is check your file formats. This is input format, and this is output format. So in the input format is where we upload. We bring in those clustered ligands of aspirin derivatives. Then we keep this format in the same format that they were downloaded. So we downloaded them as SDF. So we maintain here as SDF. But the output format, we want to convert them to SMILE code. So that means we'll change this output format to SMILEs. So kindly come to this output format. Look at my cursor, this drop down arrow. And below SDF, you see SMI SMILE format. Can you see it?
Yes, I can see.
SMI. Don't pick SMILEs. Don't pick the SMILEs. There are two SMILE formats to use the SMI SMILEs format.
Yes.
Fine. So once you click on that SMI SMILEs format, it means that whatever will be generated as output will be in SMILEs code, will be in SMILE format, while our input is left as SDF. So we are going to now import that download under this. Yes.
Hello, sir. Before we continue, sir.
Yeah.
In the OpenBabel, in the input, we have SMI there already. Are we going to change it to that SDF before we continue?
Do change it to SDF.
Okay. So that we
Yes.
leave the output as SMI smile.
My format. Good. Yes.
Okay. All right.
Yeah.
Sorry, sir.
Because, yes.
I saw SDF MDL MO. Is that the format?
Yes. SDF MDL MO format. Yes.
All right.
Yeah. Oh, thank you. All right. So, are we good to go?
Yeah.
We are good to
Okay.
go, sir.
So, you can see beside this white box, there are 3 dots, 3 horizontal dots. Look at where my cursor is pointing at. There are 3 dots there. So we shall click on that 3 dots. That will open up our file folder. Our folder. We now choose the folder where we have the compound. Then we lift it up. So click on these 3 dots. Once you click on that 3 dots, locate the folder where go to your download. Mine already opened my download. Then look for what you downloaded. PubChem search record. So once you look for PubChem search record, click on it and click open. So once you click on it and click open, it will be opened here. These compounds are too large.
Yes,
my
my
system not
own
crash.
is open already. I can see 000 with some numbers.
Fine, good. I'm just betting on my system. Let it system. Go ahead.
Hello.
Yeah,
Please,
yeah.
sorry, I didn't quite get this last one. When you clicked on the 3 dots, what next?
Yes.
Please.
When you click on the 3 dots, it will open up a folder. Like so to locate your download where you downloaded that compound. Because once you have downloaded that our analogs, then to be saved in your download. So go to your download. Then on your download, click on the file that was downloaded.
Okay.
Then click open. Then click open. So once you click open, you will see some 00 stuff showing in this empty space.
Okay, sir.
Yes. Okay, well, let me give you the procedure why mine is still opening. Let me give you the procedure so those whose desk has opened can continue. I think
Sorry, you are talking about 000. Where is that 000?
Yes, if you have clicked on open, it will open up on this. See where I move my cursor, this empty space. It will be something will show up here. Mine is still loading.
Mine has opened.
So forgive me, I'm running something background. I'm on the program, so it might take time. But while that is still going, I will explain what next to do, so you can finish it up. So even if it's ending at this place, then tomorrow will continue. Now the next thing you will do now is come to the output. Okay, thank God for my system. It has opened.
Sorry, my own is not. My own is not. It's not showing the download.
Hey,
I don't know. It's not taking me to the download.
Okay, but has it when you click on the 3 dots?
Yes.
What did you see?
I just see empty box. Then by the site, I click on download, and it doesn't take me to.
Fine. Look at the problem.
Okay.
Hope your input format is in SDF.
Yes. SD. Okay.
SDF.
This is SD. Yeah. Okay.
SDF, MDL, both formats.
Yeah, that's the problem.
Because what's happening is that he is looking, he could not find any file in that format that was there.
Oh, I see. I'm there.
Oh, thank
Thank
you.
you.
You're welcome. So at this point now, come to the output file. This output, there is also a 3 dots here. There's also 3 dots in the output.
Yes.
So the essence of this one is to save, choose where you want the result to appear, and then save it with what you want it to be at the title, what you want the file to, the name of the file. So click on that 3 dots. So once you click on the 3 dots, choose where you want it to be. Me, I will still want it to be in my download. So mine is showing download. Then give it a name. What do you want to call it? Aspirin derivative smile code, or aspirin analog smile code, whatever you want to call it. Aspirin analog smile code. So I called it aspirin analog smile code. You call it what you want to call. Then click on save. So what we just did now is that we have specified the location where we want the file to be saved and the name of the file. So you don't have any job again. Just click convert. At the middle of that open barrel, there is convert. This convert. So once you just click convert, what will happen is that it will convert those structures from SDF to smile code.
Where is that convert?
Look at it. At the middle of that open barrel, you see convert. The middle. Look at my cursor.
Okay.
Yes.
You are doing well,
We have
sir.
convert only if match smart. Then we have filter convert only when test are true. Which one?
Don't click anything. Just click only on that convert.
Okay, the one on top or at the top.
Yes, the one on top. Yes, the one on top.
Okay.
So just click convert.
Yeah, mine has been converted. Nice one.
Mine.
All right. How many molecules converted? Mine is showing 5,245.
Yes, the same thing here.
Yes, same as mine.
Likewise here.
So, but meanwhile, they were about 6,903. But because we focused on 3D.
3D. Okay.
So don't bother. This result has been saved already. Go to your download, you will see it saved. Go to your download, you will see it saved already. Look at my download. It's saved as aspirin analog smile code. Can you find it?
Yes, sir.
Good. It's saved as notebook file. I don't know the format of your own. Mine is in notebook.
Mine is in SM.
Mine also notebook.
Okay. Irrespective of the file format that it is, there is no problem. We can open it in Excel. We can open this result in Excel now. So open your Excel. Open up your Excel. So this is where we end. Then by tomorrow, we'll now from here to add to where we can generate the properties we use to set our model. So open your Excel. Let's just open this file in Excel. So, yeah. Have you done that? Launch your Excel. Have you done that? Have you launched your Excel?
Yes.
Okay, so do Control O for open. You can click Control O for open. So once you go to open, or you click on file, then click on open. So we want to open the folder in Excel. Can you see my screen?
Yes.
Now, go to all files, select all files, keep it at all files where my cursor is. If yours was in Excel file, change it from Excel files to all files. Are we together?
Yes. Yes, so we are together.
Me, I did not see anything in download again.
No, change it to all files.
Okay.
Yes. If it was an Excel file, click on that and you will see all files. All files, not all Excel files, only all files. So once,
Sorry, if I get it very well, you open the Excel, then
yes,
you click on
go to file and click on open.
okay, file open.
Yeah, as if you want to open, a work in Excel.
Fine.
All right. Are you there now?
Open.
So having clicked on all files, can you see this my file?
Yes, sir.
Yes.
You can see. Good. So click open. Click open. It will open up in Excel. Now, once you click open, it will show you this in my screen. Can you see it? Is it the same thing with yours?
Hello, sir.
Yes.
Yes.
I need assistance, sir. My own didn't come up with Excel. It only come with Notepad and Word.
No problem. No problem. But we'll open it through Excel.
How can I go that way?
Have you opened your Excel?
Yes.
It didn't bring forth the Excel issue. It bring forth Word and Notepad.
No, do you have Excel in your system?
I have.
Excel.
Yes.
I have.
Yes.
Open your Excel first. Open now. Launch your Excel.
Okay.
So now launch your Excel separately now.
Okay.
Because we are using Excel to open the file.
Okay, sir.
Okay.
So when you have launched your Excel, you click on file, then click on open. Click on open. You can choose browse. The video will guide you, so we are almost 5 minutes late, so the video will guide you.
All right, sir. No problem, sir.
So I will encourage you to make that commitment feel, so you can have this video for yourself. You can download and you can replay anytime. So that you just finish not having the video and having the materials. Then all files. If here is showing the file, change it to all files. Mute your mic. I can't hear myself. So keep it at all files. Once you keep it at all files now, you will see that's your Smile Code file. So click on the Smile Code file and click open. Now, once you click open now, it will show you 3 steps. Whose mic is on? Oh.
Yes, sir. Oh, sorry, sir.
All right. So it will show you this stuff now. We have just 3 steps to do. It's a very simple thing. Click on next. At this stage, click on next. Don't do anything. Just click next. Click next. Click next again. Click next again. Then click finish. So it has opened up. All right. Are we on the same page?
Yes, sir.
So
Yes.
all right. So what you can see now is you can see your smile codes and your PubChem CID. So the first one is 4133. And if you even check, if you check back to your PubChem, the first molecule there is 4133, right? On my screen. The first,
Yes.
yeah. So
Yes.
okay. Okay. So now we have succeeded in retrieving compounds. We have retrieved so much 5,345 compounds. So we want to, our aim is to screen these 5,345 compounds, screen them based on the models we set. So we shall generate the properties of these compounds using the smart code. But that will be tomorrow business and more. So save this thing now. Save it. Let it now be saved in Excel. So you can directly open it by clicking on it. It will open in Excel directly. So you can do save as. You can do save as, so that it will save as an Excel file, no longer as a notebook or SME. So you can save it back. Save this one now. Okay, so you can save this one that you have now as an Excel file. You can call it aspirin Excel smart code. Aspirin Excel smart codes. So once you have saved it now, you can easily open it now directly. Say yes. So you can easily open it now from the folder where it is saved directly in Excel by double clicking on it. So far we have succeeded tonight.
Sorry, are we saving it as an Excel file?
Yes, you can save it as Excel file now. Like save it now as Excel. You can go to save as and save it as Excel, as a main Excel file now. So for you to do that, you change this save as type. This save as type, change it from text, change it to Excel. Sorry, to Excel workbook. So change this save as type. This save as type, change it from a text. Just click on it and look for Excel workbook. Look for Excel workbook. The reason why we are doing this so that it can easily be opened now directly in Excel without stressing us anymore. So this is the file Excel. So you can see now is bearing my, is having my Excel icon. Excel aspirin Excel smart code. So I can easily open it now by double clicking and it will just open in Excel. All right, we have done a lot tonight, and we shall continue tomorrow. So this is 11 minutes after 10.
My PubChem download. Is it
Please.
or any other thing? Can any other thing cause it?
Okay, I didn't get your question. You said
My PubChem download is failing. So I
So
want
I
to
just open my Excel.
can any other factor cause it?
like to download the PubChem first, to download those analogs from PubChem.
Yes. Yes.
Okay, check your internet. That will be the only thing.
Okay, another question that Assuming we downloaded through this, and we want to convert it to it, how do we do that in open?
Okay. I will show you tomorrow, or probably by March. I don't want to start teaching Open Babel now, but we have a good module for Open Babel by March in the 1 month training. But I will show you tomorrow. I will show you. So just remind me tomorrow.
Sir, I just
Okay. Yes, it can go ahead. Let me hear you. I can't
my
hear you.
Excel, please. After clicking on
Yes.
yes, sir. Hello, sir.
Oh, yeah, I
open
can
my
hear you.
Excel lately. After clicking on the file by the left side. Okay, what will be the next thing? I click on file.
Oh, I wouldn't like to go back now.
Hello?
By tomorrow morning, you will get the video by tomorrow morning, so it can help you.
Okay. Oh, all right. Thank you very
Because
much.
that will make me to go back and start showing you. Because if I use my mouth to say it, you may not grab it. So, but by tomorrow you get access to the video. Tomorrow before 8 a.m. because we always send our videos before 8 a.m. in the morning. So you
Oh,
get the
all
video.
right. My Excel opened lately, so that was all.
Sorry.
All right. Thank you very much, sir, for this wonderful lecture and very better one. I really enjoyed it. I just wish to ask this question. Will you send the video directly to our mail? That's number one. Number two, how do we do the payments, any account details, and any need to send evidence of payments? Thank you.
Okay. In the mail that was sent for this training has a payment link. So in that mail where you got the meeting link, the download link, if you read down, there is a payment link. So once you click on that payment link, I say click here to pay now. Once you click on that payment link, then it will open up the Google form. On that Google form has the account details. So once you pay to the account details, then fill up the Google form. Yes, once you pay to the account details, then come back to the Google form, fill your email, your name, just your email, your name, and then upload the payment receipt. So you can screenshot or download the receipt, upload it in that form and submit. So it is the emails of those who submitted that form will get the email from there, then send the Google Drive link of this training to that email. Those email that made that payment. So no longer general email, because you are just making that commitment fee to assist us, because it's a free training and we are not charging you. So you're just committing the payment for processing miscellaneous and getting access to the training materials. So in that mail that was sent to you for this training, the payment link is there. Thank you very much.
Yeah, thank you. I hope it's 2,000 naira or
yes, 2,000
okay.
For Nigerian. Then our international participants, they are paying $3 USD.
All right, thank you very much for the clarification.
So except you're in Nigeria and you still want to pay the $3 USD.
That sounds funny. Thank you very much.
Yeah, thank you. All right. Do you have any other question? Okay, I've answered the Obo Chioma Messi. I've also answered the question about the payment.
Yeah. Sorry, I'm not talking too much. The training tomorrow, is it the same 8 o'clock?
Yes, the same 8 p.m. what?
All right, thank you.
Yeah, thank you. Okay. We have tried so much to stay to this point. On behalf of Hendeb Industries Nigeria Limited, we say thank you for coming around. For our first timers, the first people who is attending our training for the first time, we have a culture. So just bear it in mind as we close this meeting. We have a culture in Hendeb that if Hendeb must train you, whether a free training or a paid training, if Hendeb must train you, then Hendeb cannot give you certificates without you showing Hendeb something you learned from that training. Now, because we are paying 2,000 for commitment, which include the certificate fee, if you do a task, because we'll give you a task, we'll do a mini project based on what we taught you now. We are expecting that you do a mini project. So if you finish that mini project, we'll give you certificate of completion because you completed a task. But if you choose not to do a mini project, they will give you the certificate of participation, which means you participated in the training. I will encourage you people to engage in the mini project. From what you just did now, choose what you want to do. Just choose a word. Like we use aspirin. You can say to work on maybe a natural product by tomorrow training, or you say to work on any heat or any drug you want to modify. You can choose that drug and just do a virtual screening. At the end, tell us that from 5,000 compounds, we're able to select this 50 or this 10. And we're okay. We have seen that you have done something for what we taught. We are not after a paper. The only time we ask for paper is in our monthly training, that you must have a publication for yourself, not for us, for yourself, before we can issue you a certificate. But for this free training, except you want to settle for the certificate of participation, which means you joined in the training and you participated, which I don't like. I prefer certificate of completion. So we'll be given 2 weeks after today, after the training, 2 weeks to finish up our mini project. So by the end of tomorrow, I will drop the guideline for the mini project. I will encourage you to do that. So thank you very much as we retire to our bedside.
Thank you,
Thank
sir.
you,
I
sir.
later got it. I got it perfectly.
Thank you very much. Thank you very much, sir.
If you're not in the WhatsApp platform, please, you can signify or send me a DM. So I will add you up to the WhatsApp platform. Very important. Okay, so good night.
Thank you so much. Good night.
All right, good night,
Those
sir.
in Yoruba, how do you say good night in Yoruba?
Thank you so much, sir. Good night, sir.
Oda
Ọ̀dàrọ̀.
Oh, Odaro or Idaro. Anyone?
Ajilayo.
Ọ̀dàrọ̀.
Ajilayo.
Okay, Odaro. Oh, Atiyala.
Ajilayo.
My dear, I don't want to bite my tongue. Odaro, good night. Thank you. Esther, how do you say good night in Kenya?
We speak English, Swahili, and local languages. Which one do you want me to say?
No, local language.
Oh, local language. Yes. I come from the same language as Barack Obama.
But
we say,
okay.
Yeah, we say need my bear.
Need my bear.
Yes.
Need my bear.
Yeah, need my bear.
Okay, bye-bye.
Yeah, bye-bye.
Lady Mabel.
I hope you received my own submission.
Yes, I received yours. Maybe na bad.
It's my
Oh
bad.
my God. Maybe na bad. You too.
Kachifu, sir.
Kachifu. Good night.